Hello, my name is Jonathan Cherry. I am an assistant professor at the Department of Pathology and Laboratory Medicine at Boston University School of Medicine, also a research health scientist at VA Boston. Um, Today, I'm going to be telling you a little bit about our project looking at the evolution of different isoforms of tau in chronic traumatic encephalopathy, or CTE. So this is a project that is using the opal staining platform as well as the Vectra Polaris to really get at multiplex immunofluorescence in tissue that is quite difficult to work with in terms of autofluorescence and lipofussion, and look at some immunoprofiling of a couple different neuropathologic markers that are of pretty important interest to us in just overall disease. It's important to note that all the views expressed here are going to be my own, and I have nothing to disclose. So let's get into it. So this project is looking at tau pathology in neurodegenerative disease. So there are a lot of different tauopathies. Tau is pretty ubiquitous among a lot of different cells in your brain. It is a microtubule-associated protein that's enriched in neuronal axons. It's really important in stability and axonal transport and support. But under different conditions, it can become unbound and it becomes pathologic, where it can be hyperphosphorylated and begin to aggregate together. And that's where you get this pathological phenotype, is when you get these unbound hyperphosphorylated aggregates of tau. And it's a hallmark of many different diseases. So it's not just present in one or two diseases. There's a whole class of different tauopathies, and there's a bunch of different acronyms and letters down here to show that off, that typify tau. They're all termed tauopathies. But they're all not the same. They all share different features of this tau based on many, many different things. So all tau is not the same. It can undergo different sorts of hyperphosphorylation states. So this is the MAPT protein, the tau protein from the MAPT gene. And there are a host of different areas where this protein can get hyperphosphorylated once it's unbound, and that can lead to very different unique conformational structures, pathological variants, modes of activity, and appearances and aggregation states of tau. And this is just a small sample of the different states that it can be phosphorylated. So tau is quite different in that way. Additionally, there have been several studies that have looked at the conformational structure of tau itself among different diseases, and the 3D conformational structure, looking at the crystal structures, can be quite different, and even in these very related diseases. So again, just because it's phosphorylated in different areas, the structure of the tau itself can be quite different. Additionally, and more relevant to this talk, there's a lot of differential splicing that can occur in the MAPT gene seen here. So the MAPT gene can undergo splicing in several different areas. We're more focused on exon 10 and what happens with exon 10 when it's differentially spliced. It can lead to 6 different isoforms of tau. So this is on top of all the other things I just mentioned, different conformational structures, different hyperphosphorylation states, and then you can have this alternative splicing, which just makes it quite complex to think about. You have these 6 different structures of tau. And collectively, based on the inclusion or exclusion of exon 10 here, you can term them 4R tau, because they have 4 repeats when they have exon 10 included. And then without 10, they have 3 repeats. We call them 3R tau. So we have 4R tau and 3R tau, which is pretty biologically relevant. It's not just different splicing, and they do the same things. Go forward. So 3R tau and 4R tau are pretty biologically relevant. 3R tau is more present in development. As you develop further and into adulthood, you get even amounts of 3R and 4R. And different diseases seem to be typified by 3R and 4R in different quantities. So, and these are all tauopathies. So there are 3R predominant diseases, so they only have this 3R phenotype present. There are 4R predominant diseases, and there are 3 and 4R prominent diseases, so they have both. So this has become a way that you can identify different diseases based on just this neuropathological staining. And it's become quite useful. And it really shows how if you better want to understand, or why we should better understand 3 and 4R across different diseases, because it's possible for better diagnosis and biomarkers, which is a really important aspect in neurodegenerative pathology, is how can we find biomarkers and how can we identify these things? Because many of these diseases you can't diagnose till after death. We're getting a lot better at that with a lot of them, but many of them you have to wait till the person has died, do an autopsy and actually look in the brain and then say, yes, they had Alzheimer's, they had CTE, they had PART, all these different diseases. So there's kind of a rush right now to really try to understand, can we find different targets in the brain that we can identify someone in life? And that starts with the better understanding of neuropathology. So the disease that we are focused on is chronic traumatic encephalopathy. So CTE is the disease that is associated with repetitive head impacts. So this is received through playing contact sports or military service. So this is if you're ever watching a football game and someone's talking about the concussion disease, or he has so many concussions, or he has a lot of head trauma, which can lead to problems later in life. This is what they're talking about. My big spiel whenever I give this talk is that this is not a concussion disease, even though everyone likes to call it the concussion disease. This is the disease where it's the exposure to repetitive head trauma. So the hits that the athletes or people in the military get every single time they serve or they play a game, over every single practice. It's the subconcussive hits that are really driving this pathology that we see. Now, there's been work in the past couple of years to identify what CTE looks like neuropathologically, and I really discovered the pathognomonic lesion that it's called. So this is the feature of the feature that you can see neuropathologically that defines the disease. And it's this perivascular accumulation of tau. So perivascular means it surrounds a blood vessel. And again, this is tau pathology. And this pathology is found early in the disease at the depth of the cortical sulcus. So your brain has folds in it. At the bottom of the folds in the cortical gray matter, you get this accumulation of tau perivascularly. And this is unique to CTE. You won't see it in any other neuropathology. And it defines the disease. So CTE can be split up into multiple stages based on where this tau pathology is found. In stage 1, you get these 1 or 2 of these isolated clusters of this perivascular pathology in the frontal cortex. Stage 2, you'll get more of these clusters of tau throughout your frontal cortex, potentially parietal cortex. Stage 3, you'll have more tau, but you also start to get stuff in your medial temporal lobes, so your hippocampus and amygdala start to be affected. In stage 4, it's typically brain-wide. Your cerebellum can typically be spared, but you'll still see it in very severe cases. We tend to group these into binary groups now. So you have stage 1 and 2, we kind of cluster as low stage CTE. Then stage 3 and 4, we cluster as high stage CTE. So it's really of interest to better understand this spread. And there's been some early work looking at the tauopathies that are present in CTE. And we've shown previously that there potentially is both 3R and 4R isoforms present in the brain in someone with CTE, but this was very limited at the time, and what this study is going to be doing is expanding on that literature, because this doesn't tell us too much about the neuropathology as it stands. There's never really been an in-depth characterization of the 3R or 4R isoforms in CTE. The previous figure I just showed you is a very, very small sample set. So that needs to be shown over multiple cases and looking at all these stages of pathology, not just looking at very severe disease, which is typically focused on in these studies. So we need to have more cases. So again, this needs to be in the dozens, if not hundreds of cases to really understand the neuropathology. We need to have biochemical confirmation, so take it beyond just histology, look at biochemical staining. And really importantly, we need to do multiplex immunofluorescence of this. So as I just mentioned, this has all been done on serial sections, and it's never really ever been looked at least in CTE, all on the same section to look at 3R and 4R and our tau stain here, which is 88. It stains all tau all together, which I think is really, really, really important. And that's something that this was one of the goals of this study, was to do these high multiplex stains in one piece of tissue, like I think was mentioned before in one of the previous talks, to get as much information out of one case that we can. So the methods for us doing this are going to be using postmortem tissue from 99 contact sport athletes from the VA-BUCLF brain bank. This is a brain bank present at Boston University. It is the largest CT brain bank in the world. We have over 1,000 cases in there right now. We use 99 individuals that all played contact sports, and the majority of them played American football at high levels. So these were individuals who played in collegiate or the National Football League. So they all have significant exposure to head trauma, and they have a neuropathologic diagnosis of CTE using the criteria that we talked about previously. So they have a neuropathologic lesion. And importantly for these kind of studies, they didn't have any other comorbid disease. So they didn't have a diagnosis of Lewy body disease, Alzheimer's, FTLD, motor neuron disease. They had just CTE. So we're trying to better understand how the tau profile looks in this disease. We don't want to have any other diseases present that might confuse us and kind of cause contradictory results. So Alzheimer's can all these diseases can occur with CTE. So it's important for these studies just to have CTE alone. We're going to be looking at the dorsolateral frontal cortex. So this is the area of the brain where pathology is first, one of the areas where pathology can first begin. We thought this would be a more sensitive measure of early changes and let us really get a true spectrum of the overall phenotype as it progresses from stage 1, 2, 3, and ultimately end stage. These are going to be looking at whole slides for our analysis when we finally get there. So we're going to be looking at both gray matter and to some extent white matter, but majority gray matter here. And we're going to use two different techniques. We did a biochemical analysis, which I will go through quickly because I don't think that should be the focus of this talk. Then we did a histologic analysis, and this is what I think people have a lot more interested in and for this particular project. So this was quantitative immunofluorescence at the dorsolateral frontal cortex at the depth of the cortical sulcus. Initially, we used 3 different antibodies. So p-tau, which is going to stain all of the tau, that's hyperphosphorylated and pathologic. A 3R tau antibody and a 4R tau antibody, and these are all going to be done in the same tissue. And then I'm not mentioning it here, but we're going to be doing a 7-color stain at the end using these antibodies plus 4 others to look at tau in different cell types, which I think is kind of where this project is going. So this is a project that is using the Vectra Polaris to spectrally mix and deal with the background issues in lipofuscin commonly found among human tissue. And I think that's really important to point out that we're working with tissue that can be quite old. So our brain bank has tissue that is 10 to 15 years old. So some of these cases are quite old as well, up to 90, 100 years old. So they generally have a lot of issues with background and non-pathologic protein aggregates that are quite fluorescent, which really hamper a lot of human neuropathology analysis with immunofluorescence. So part of the goal of this project was to develop techniques where we could better look at human neuropathology in these heavily fixed old age samples to find markers that can let us do these multiplex stains. So first, I'll just quickly go through the biochemistry, which is an important part of this study that I don't want to just kind of drop off completely, to look at the overall levels of tau and 3R and 4R. So we ran just basic immunocyte Western blot analysis, looking at the overall amount of tau, and we can look at the different aggregates of the isoforms based on their size on a Western blot. And we can see that when we quantify them, there is an overall decrease in total tau and decrease in monomeric tau, but an increase in total tau. And what this is showing us is that there is unbinding from tau from the microtubules, and when it's unbound, it becomes these monomers. So you're getting monomers that are then unbinding, and then they are aggregating together and becoming oligomeric. So you're losing the monomeric pool and increasing the oligomeric pool. So this is just showing us that, yes, there is tau unbinding in CTE, and you're getting this oligomeric pathologic incorporation of them overall. We also looked for the amounts of hyperphosphorylated tau, this is pathologic tau, and like we've seen in previous studies, you see an increase in pathologic tau according to CTE stage, which again isn't surprising. We do the same thing with 3R and 4R specific antibodies, and we see very similar changes where there is this increase in increase in oligomeric 3R and 4R tau. And what this shows us pretty much off the bat is that CTE is a mixed 3R, 4R tauopathy, which was suggested before, but it's really never been shown in depth and through this technique. So we're pretty sure that CTE does have 3R and 4R. That is typified of the disease, but something that we can't get at is in this biochemistry setting is progression specific to the CT lesion. So, biochemistry is great for looking at these specific changes, but you need to look at the tissue itself with IHC or IF to see these changes directly around the blood vessels, which are typified of CTE. So that's how we know what CTE is neuropathologic lesion. So we wanted to use our immunofluorescent background to better understand it in CTE. So that's what we did. We did multiple different stains in the same tissue, looking at AT8, 4R tau, and 3R tau. And off the bat, what we see is that CTE is a 3R and 4R tauopathy. You have equal levels, at least in this image here, of both 4R and 3R. And we wanted to actually quantitate this because that's where the strength of these stains really come in. We can actually get quantitative data through physically counting the amount of 3R positive and 4R positive cells. This was all done by hand by me originally, but we are working on techniques that are allowing for automated counting of this. One of the downfalls of working in neural tissue is you have a lot of irregular shaped cells. So neurons, astrocytes, microglia, they don't really follow a set pattern here. So it's, you can see it here, you have all of these positive neurons here that are all quite different. So it's hard to get automated methods to count them. So the original analysis here was all going to be done by hand counting. But we're working with AI right now to better quantitate these so I don't have to spend hours at my desk counting these and do more productive things. So we wanted to quantitate the amount of 3R and 4R based on this histology over CTE stage. We know it's going to go up, but we don't know specifically about it in the different aggregates and different stages of the disease. And what we see is very similar to our biochemistry. We get an increase across CTE stage for 3R and an increase with 4R. So this is very similar to our biochemistry, but again, what this doesn't tell us is the individual contribution in one tissue. How do these look when you compare them overall in case to case when you do these, you know, what's going to be here is a 3-plex. Can we determine different profiles based on our analysis? And what we end up seeing is a couple different phenotypes when we did this multiplex. The first is when looking at mild CTE, you can see that this is your neuropathologic lesion right here. You have this 4R predominant lesion. Even though there's 3R there, it's mainly 4R. So this is a disease that, at least in early stages, seems to have primarily 4R. When you look at severe CTE, you do see 4R predominant cases. Again, they have 3R, but they're mostly 4R. But what we end up seeing is that there is another subset of individuals that have a 3R predominant lesion. Again, they have 4R present, but through the immunofluorescence, we can see that the majority of tau present is in fact 3R. So there's different presentations within the same disease. Again, all of these have, if you look by AT8, the neuropathologic lesions, so they're all CTE. We wanted to quantitate this now, so we developed a ratio by pretty much counting the amount of 3R and 4R positive cells there, and we divided the amount of 4R cells by 3R cells, just a very crude ratio analysis. And what we see is that this is log transformed to show the spread. There is an increase in the ratio of 4R to 3R, so there are more 4R predominant cases in very early stages of disease. But once you start to get to stages 3 and 4, so very severe disease, you start to get a dip into more 3R predominant cases. And when you actually use the amount of tau staining as a surrogate of disease severity, you see there's this negative correlation. So you're getting a change from this 4R predominant phenotype to a 3R predominant phenotype that seems to change with disease severity, which is, I don't think it's really been shown before. So that's a really important thing to kind of get at now. Another important analysis in this is to really understand what cells are containing tau. It's good to say that 3R and 4R are there, but another important feature in neuropathology is understanding which cells contain tau. Are they neurons that contain tau? Are they astrocytes that contain tau? Because different diseases can be typified by either neuronal or astrocytic aggregates of tau. So we need to better understand that. And the way we initially did this was, again, by a hand method, using neuropathologists who can recognize astrocytes and neurons based on just the tau staining itself. So they have a very, very specific appearance, and we use neuropathologists who are trained to identify this to look at blinded sections and pretty much count astrocytes versus neurons. And then we can examine the total amount of tau-containing neurons and astrocytes around the lesion, and we can determine another percentage. So the amount of neurons or astrocytes divided by the total tau-containing cells, and we can see, are they more 3R or are they more 4R positive with each of these phenotypes and how that might change over time. And this is what we can end up seeing. So pretty much what this shows is that only neurons contain 3R tau. So only neurons will contain 3R tau. But astrocytes can contain both 4R and neurons and astrocytes contain both 4R tau. What the difference between these two groups is, in severe CTE, 4R tau is found in astrocytes only in this later stage. You typically don't see astrocytic 4R tau in early stage CTE. So the early CTE lesion, neuronal, and over the course of this disease, it becomes more astrocytic. One thing that segregates, this late stage CTE or high stage CTE from the early stage CTE or low is that these individuals tend to be much older. So we figured that this might be a product of age and not pathology. So we plotted all of our cases by decade of age of death and did the same sort of ratio analysis. And what we end up seeing is that you only start to see astrocytic tau, 4R tau. This is all 4R tau because that's, only astrocytes have 4R tau. You start to see it emerge about 60 to 90 years old, or 60 to 69 years old, and it only increases from there. Now, this isn't due to some cases just having more tau than others. What you typically see is that there's several cases in this young age range that have very severe tau pathology, but it's all neuronal. You only will see astrocytic tau in later stages of disease. So we find that age is really driving astrocytic 4R tau. Now, the last thing I wanted to do here is follow up on this morphologic assessment. So the previous results I just showed you were primarily based off of morphology. And this is not always ideal to examine in a quantitative measure because some cells are really hard to determine. Are they neurons or astrocytes? And sometimes, depending on who you ask, what neuropathologist, it could be different. They might have different ideas on what exactly a neuron should look like, what an astrocyte should look like. And this is going to cover every single cell. There's going to be cells that we can't quantitate. So this really shows the need for this high-level multiplex immunofluorescence. We need to stain for multiple different markers of cells, such as astrocytes, neurons, other glia, such as microglia, macrophages, potentially oligodendrocytes, in the same context of tau, 3R tau, 4R tau. And so that's what we did. We did a multiplex immunofluorescence. This is going to be a 8-color stain, 7-color with our merge down here. Looking at multiple different markers taken at the depth of the cortical sulcus. So this is a very shrunken down version of a whole IF slide that we did with again, using the opal system and Vectra Polaris to deconvolute and spectrally unmix, not deconvolute, just spectrally unmix this image. And what we end up doing is doing our 4R tau stain, our 3R tau stain, and our overall tau stain, coupled with MAP2, which is a stain for neurons, GFAP, which is a stain for astrocytes, IBA1, which is a stain for microglia, and DAPI, which is just going to be our nuclear stain. And what we end up seeing, again, this is a case that has severe pathology, and it's quite old. So we see that when you look at the amount of 4R tau, these white arrows, you do see 4R tau in neurons, and then you see 3R tau also in neurons, again, these white arrows here, but you also see 4R tau in astrocytes. So we're showing that 3R tau and 4R tau exist in both, or I guess neurons are going to be more 3R tau, but you're going to get 4R tau in neurons and astrocytes, which is validating the morphologic data that we did, but this is also expanding on it. And this is something we want to take into future studies, is this high-level non-morphologic dependent assessment, and develop techniques that are going to be able to quantitate this for us without spending hours at a computer screen counting these things, because now we have the power to look at all of these different markers in the same tissue to do this high-level cell type analysis, which I think is going to be really important for neuropathology in the future, which really it takes a lot of the subjectivity out of it, because we can say for sure that this astrocyte here contains tau, and we know this is an astrocyte because it stains with GFAP, and we know this is a neuron that contains 3R tau and 4R tau because it stains for MAP2, and all the guesswork is taken out of it. So with that, overall, what I've shown is that using quantitative biochemistry and immunofluorescence, we find that CTE is a mixed 3R, 4R tauopathy that evolves over the disease. It begins as a 4R predominant phenotype and then transitions more towards a 3R phenotype. And then neuronal tau is strongly correlated with CTE pathology, while astrocytic tau is more correlated with age. And if you want to see this paper at all or read more into it, it was recently published, and the citation is down here. And with that, I have to thank everybody in the BUVA CTE Center, Dr. Ann McKee, who runs the center and is my mentor, has been very helpful on this project in terms of the direction and helping with understand the neuropathology. Dr. Torstein, Dr. Hubert, Dr. Vickert Alvarez, Dr. Jessamez. There's a lot of people. This is a small list. My colleagues at Mount Sinai, Sun Ho Kim, which is a co-first author on the paper, and Dr. Jon Cherry, who is a co-senior author, and Kirk Balfour Weiss, and of course my funding sources. And with that, I can take any questions.
Thank you very much, John. That was great. Not just really everything. I was nerding out with the science, considering my background in CTE and traumatic brain injury, but the images and everything. And we always get the question of, do you need multispectral imaging for neuroscience? Is it, how do you do it? Does it work? Well, this is the perfect example of how it works, and the work was great. So thank you. Do we have any questions? We do have one question that asks, are there any differences in tau reactivity of the various antibodies in FFPE versus frozen tissues? Yeah,
great question. Most likely going to be yes. We've really done most of our work with FFPE tissue, since that's kind of the majority of what we have. We're getting more into frozen right now. We do know there are differences in the antibodies, so they have to essentially be re-optimized again in frozen tissue. So I don't have a great answer for it yet, but it's something we're working on a little bit more. But again, most of our tissue is fixed, so we typically focus on that for these initial analyses.
We have another question. Did you use Codex for the antibody stain?
I did not. I did the opal protocol.
Take one more question. Post-mortem tissues for brain, how do you QC the tissue is good or not?
Generally, so these analyses are taken after they've gone through their clinical or their neuropathologic workup. So we'll know if they've stained well or not for a lot of the clinical markers. It's tough to know if the stain is minimal, just because there's less stain or it's due to a long PMI or postmortem interval. We do have the postmortem interval on every case, so we run that as a covariate analysis to see if it determines any sort of significance on the results, but it's tough to say. Human tissue is pretty variable in general. So even though two cases are they stain well, they can stain slightly differently between each other. So our technique is to use kind of a shotgun approach and try to stain as many cases as we can to capture the variability between human tissue, because it's a challenge to work with this sort of tissue, just because it is quite, you do have a lot of variability. So, the more cases we can use, the more we can rule out variability. And if one case does have some, not great staining to it, we can kind of make the decision, is it really in line with everything else or not?
We have a quick follow-up to that. How long before tissues were fixed? Hours, days?
It's pretty variable too. So we are a brain bank that works off of donations, and we don't ever solicit them, so they kind of come to us, and it could be anywhere from a couple hours to sometimes days. We typically don't take cases that are longer than 48 hours, but we have had pretty good staining success in cases that have a postmortem interval. So the time they die, the time they are fixed at 48 hours. So we generally don't see a drop off till maybe like 72 plus hours, but we have been known to take cases that can have many days, not many days, but several days of a PMI. Just because we're a brain bank first, and then we do research with the cases that come in after they've gone through neuropathologic assessment. So we don't always have the luxury of saying, well, we're only going to use cases that have a 2-hour PMI.
And I'm going to ask one more question that we have coming in, and really this is more for selfish reasons because I get asked this question all the time with prion work. Can you speculate on how the deposits travel in the brain with CTE disease progression? De novo deposits with new trauma or prion-like dissemination?
Great question, and I wish I knew the answer to that question. There's probably a bunch of different techniques that this is propagating. I do think part of it is through a prion-like mechanism. So the connections that we see in CT are pretty similar to those in Alzheimer's. So Alzheimer's, you typically think of tau pathology beginning in the base of the brain and working way up. CT is different. It starts at the top of the brain and works its way down. but the regions that are affected are pretty similar. So we think the same sort of idea that Alzheimer's is spread through, interconnected neural regions. CT also uses those regions, but kind of in reverse. So you do get this neuronal, neuron to neuron spread via potentially a prion-like mechanism, but we also know that there are other mechanisms such as exosomes secreted by microglia, which can phagocytose this extracellular tau, and it can spread to distant brain regions that way. So I think it's going to be a combination of several ways it's spread.
Yeah, even with prions too, right? It doesn't always make sense with some natural cases. You see it, you don't see it in the gut, but you see it in random places in the brain, and it just doesn't quite click. Yeah.
Yep.
Okay. Thank you very much, Jon.
Thank you.